On October 23, 1997, a team led by Michael Caterina and David Julius published a discovery that changed how science understood pain. Nature. They had isolated a receptor from sensory neurons that reacted to capsaicin, the molecule that makes chili peppers burn. But when they heated the cells, the same receptor opened again. The pepper molecule and dangerous heat were pressing the same biological alarm. That receptor became known as TRPV1, and it immediately revealed something strange about every mouth that has ever faced a chili pepper. Your tongue is not actually detecting spice as a flavor. It is activating a neural system built to warn your ancestors about damaging heat. The burning is an illusion with a purpose. There is no flame, yet your nervous system behaves as though something dangerously hot has arrived. And people experience that illusion very differently. One person calmly eats a pepper that leaves another sweating, coughing, and reaching for milk. It is tempting to explain that difference with one sentence. Some people were simply born with better genes for spicy food. There is truth inside that idea. Human TRPV1 varies genetically, and some variants can alter how strongly the channel responds to capsaicin or heat. PubMed. But the deeper story is more surprising because DNA is only the opening move in a biological system that can be trained. Your inherited pain machinery sets part of the starting point. Exposure, culture, expectation, and nerve adaptation can then rewrite the experience. So the ancient genetic code behind spicy food tolerance is real, but it was not designed for chili peppers, restaurants, or culinary bravery. It existed because an animal that could not distinguish harmless warmth from tissue-damaging heat had a serious survival problem. Long before humans cultivated peppers, vertebrate nervous systems were already evolving molecular gates that detected dangerous temperatures. TRPV1 became one of those gates, a microscopic protein embedded in sensory neurons, waiting for heat, acidity, inflammation, and certain chemicals. Capsaicin did something evolutionary ingenious. It learned to fit a lock that animals already possessed for sensing danger and heat. The pepper did not invent our burning sensation. It exploited an ancient warning circuit and turned that circuit into a defense. Yet humans eventually performed the strangest reversal of all. We began seeking out the signal that evolution had built to make us withdraw. To understand why, we have to separate three things often confused in everyday talk: sensitivity, tolerance, and preference. Sensitivity is how strongly your sensory system responds. Tolerance is how much stimulation you can handle. Preference is whether you enjoy it. Genes can influence the first. Repeated exposure can reshape the second. Culture and psychology can transform the third. That is why people with similar biology can end up eating completely different diets, while different backgrounds can converge on the same love of heat. The story begins at the level of a single nerve ending. In your mouth, branches of the trigeminal nerve monitor chemical irritation, temperature, pressure, and pain. When capsaicin reaches those nerve endings, it binds to TRPV1 channels in the cell membrane. The channel changes shape and opens. Positively charged ions, including sodium and calcium, rush into the neuron. The electrical state of the cell shifts, and the neuron begins firing. That signal travels toward the brain as nociception, the neural encoding of potentially harmful stimulation. The brain interprets it as burning. This is why chili heat behaves differently from sweetness or saltiness. Sweet, salty, sour, bitter, and umami are classical taste qualities detected through specialized taste cells. Capsaicin belongs to chemesthesis, the chemical activation of touch, temperature, and pain pathways. Menthol creates an opposite trick by activating cold, sensitive channels. The result is sensory theater. A chili can make the mouth feel hot without increasing tissue temperature to the level implied by the sensation. TRPV1 is perfectly suited to this deception because capsaicin and actual noxious heat converge on the same molecular doorway. When Caterina and colleagues cloned the receptor in 1997, they found that temperatures in the painful range could activate it. Later experiments in mice lacking functional TRPV1 showed reduced responses to certain noxious heat and inflammatory heat pain, confirming that the channel was part of a genuine protective system. Nature. That immediately destroys the simplest version of the spicy gene story. TRPV1 did not evolve so humans could decide whether salsa was mild or brutal. Its older job was survival. It helped sensory neurons detect conditions that could damage tissue and helped the nervous system organize withdrawal, avoidance, and protective responses. A hand approaching a flame in a mouth filled with capsaicin can therefore activate overlapping molecular machinery for completely different reasons. One is responding to genuine thermal danger. The other is being chemically fooled by a plant molecule. The reason that molecule exists leads away from humans and into the evolutionary battle taking place inside wild pepper fruits. Capsicum plants evolved in the Americas. Long before domestication, wild peppers faced a basic reproductive problem. Their seeds needed to travel without being destroyed. Many mammals are poor partners for this job. Rodents and other seed predators may chew seeds, damaging the embryo and reducing the plant's chance of reproduction. Birds can be better dispersers. They swallow fruits and carry seeds away, often passing them without the same destructive chewing. Capsaicin helps create a remarkable filter. Many mammals strongly detect it through TRPV1 and avoid pungent fruit, while birds are far less sensitive to capsaicin. Experiments and comparative work have shown that differences in the avian TRPV1 channel make birds relatively insensitive to the burn that mammals experience. PubMed Central, PMC. From the pepper's point of view, that can bias consumption toward animals more useful for seed dispersal and away from destructive consumers. There is also evidence that capsaicinoids can help defend fruits against microbial attack, adding another evolutionary pressure that may have favored pungency. The crucial point is that the pepper's chemistry evolved for the pepper's reproductive interests, not ours. Then humans entered the relationship and behaved unlike almost every mammal the plant had been trying to discourage. Instead of retreating from capsaicin, people collected pungent peppers, transported them, cultivated them, selected them, traded them, and built entire cuisines around them. Archaeological and genetic evidence places the domestication history of capsicum deep in the pre-Columbian Americas, with domesticated chili peppers present thousands of years before European contact. Research on Capsicum annuum points strongly toward Mexico as a major center of domestication, while other cultivated capsicum species arose through separate histories elsewhere in the Americas. After the Columbian Exchange, chili peppers spread with extraordinary speed through Europe, Africa, and Asia. Plain ass. Within centuries, a New World fruit became so deeply embedded in cuisines from India to Thailand, Sichuan to Korea, that it can feel as if it had always been there. But chili peppers could not have shaped the original evolution of the human TRPV1 system, because humans inherited that heat-sensing machinery long before our species encountered domesticated chilies. The receptor is ancient. The cuisine is recent. This matters because it changes the direction of causality. Your genes were not selected because your ancestors needed to tolerate vindaloo. Rather, an ancient pain receptor created a sensory vulnerability that capsaicin exploited, and human culture later learned to manipulate that vulnerability. Long before molecular genetics could measure receptor sensitivity, pharmacists tried to measure pepper heat through human perception itself. In 1912, Wilbur Scoville introduced an organoleptic test based on how far a pepper extract had to be diluted before tasters could no longer detect its pungency. Modern laboratories use chemical analysis instead, but the Scoville scale still captures an essential truth. Heat is ultimately experienced through biology. Still, people do differ biologically, and researchers have spent years testing whether those differences can be traced to the genome. A 2018 study examining human TRPV1 variants found that different single nucleotide polymorphisms could alter responses to heat and capsaicin in experimental systems. One variant, for example, affected capsaicin sensitivity, while another influenced heat activation, demonstrating that small changes in the receptor can change how the molecular gate behaves. Other studies have linked naturally occurring TRPV1 variants with differences in pain responses, capsaicin-evoked cough, or sensitivity to irritating stimuli. In that Japanese study, 6 TRPV1 polymorphisms were associated with burning pain sensitivity and 11 with capsaicin sensitivity. The homozygous I585V variant was significantly associated with higher capsaicin sensitivity. But even those TRPV1 associations do not imply a single spicy food gene. Human response to capsaicin is a complex trait built from multiple biological pathways across multiple levels of the nervous system. PubMed. TRPV1 can influence how the burn begins, but the final experience also depends on nerve density, signaling pathways, inflammation, oral anatomy, taste perception, learning, and context. Even the same person does not experience the same chili identically every time. Temperature, concentration, food composition, previous exposure, and the condition of oral tissues can change the sensation. But the more profound adaptation happens inside the sensory system itself. Expose TRPV1-bearing neurons repeatedly to capsaicin, and their response can diminish. This phenomenon is called desensitization. At the receptor and neuron level, calcium entering through activated TRPV1 contributes to regulatory processes that reduce subsequent responsiveness. With intense or repeated exposure, capsaicin-sensitive nerve endings can become temporarily less reactive and the perceived burn can decline. Human studies consistently find that frequent chili consumers often report less burn from a given capsaicin stimulus than infrequent consumers. Controlled experiments have also shown that repeated low-dose oral capsaicin exposure can produce measurable desensitization. This means that part of what people call high tolerance can literally be acquired. PubMed Central, PMC. A person who once found a sauce unbearable may experience it as moderate after weeks of repeated exposure, even though the genome has not changed. The nervous system has adjusted its response to a recurring stimulus. That adaptation exposes the central contradiction in the title. Spicy tolerance can be influenced by inherited biology, yet it is also plastic. Hardwired does not mean unchangeable. Your nervous system is built from genetic instructions, but those instructions produce a living system designed to adapt to experience. This is where culture becomes biologically powerful. Children raised in households where spicy food is routine encounter capsaicin repeatedly and often at gradually increasing levels. The food is not experienced as an isolated pain challenge. It is embedded in family meals, aromas, memories, social approval, and familiar flavors. Over time, the sensory burn can weaken through desensitization, while the emotional meaning of the burn changes through learning. A sensation that initially signals avoid this can become associated with pleasure, identity, comfort, excitement, or belonging. Repeated safe encounters teach the brain that the signal is not forecasting the same outcome as a real burn. The alarm remains loud, but the interpretation becomes less urgent. That helps explain why chili enthusiasts can experience tears, sweat, flushed skin, runny noses, and intense oral burning while simultaneously describing the meal as pleasurable. Their bodies are not necessarily failing to respond. In many cases, they are responding dramatically. What has changed is tolerance and appraisal. There is an evolutionary irony here. Natural selection generally rewards animals that take pain seriously. Ignore enough warning signals and you get injured. Humans, however, are unusually skilled at learning when a warning signal can be safely violated. Cooking itself is built on controlled violations of ancient sensory boundaries. Smoke, bitterness, fermentation, acidity, and heat become desirable when context marks them as safe. Chili peppers fit perfectly into that human talent. We do not merely inherit environments. We construct them, teach them, ritualize them, and pass the resulting preferences to children. That cultural inheritance can move far faster than genes. A chili pepper left the Americas after the 15th century and became foundational to cuisines across Asia within a historical blink. Human TRPV1 could not genetically evolve across all those populations at the same speed. Culture did the work. People planted peppers because they liked them, bred varieties with different pungency, incorporated them into local dishes, and trained new generations from childhood. The global map of chili consumption is therefore not a simple map of TRPV1 alleles. It is a map of trade routes, agriculture, empire, adaptation, cuisine, family, and repeated exposure layered over shared mammalian pain biology. Yet, genetics still matters at the individual level. If your TRPV1 channels activate more readily, a given capsaicin dose may begin as a stronger signal. If other pain-related variants amplify or dampen nociceptive processing, your starting threshold may shift again. If bitter receptor variants change how the surrounding food tastes, preference can move further. Those small differences can influence how willing a child or adult is to continue exposure. And once behavior changes, biology changes with it through desensitization. The process becomes circular. Genes affect experience. Experience affects behavior. Behavior changes exposure. Exposure changes sensory response. That feedback loop is far more powerful than the idea of a fixed spicy food destiny. It also explains why tolerance tests are poor measures of toughness. A person crying over a chili may simply have lower exposure, greater sensory responsiveness, different receptor biology, or a different expectation. A person calmly eating the same pepper may have spent years training the relevant sensory pathways. Neither reaction reveals courage. Both reveal the nervous system doing what nervous systems do, combining inherited machinery with personal history. That boundary returns us to the original function of TRPV1. The receptor belongs to a protective system. Humans can train around parts of the signal, but the system remains integrated with pain, inflammation, and autonomic responses. The ancient code is still there beneath the cuisine. The same molecular gate that lets a pepper feel hot participates in detecting real conditions that can threaten tissue. This is why the 1997 discovery remains such a powerful starting point. Katerina, Julius, and their colleagues were not searching for the gene that explains who orders extra hot wings. They were using capsaicin as a biological probe to uncover how sensory neurons detect dangerous heat, or nature. The pepper was a tool for revealing something older than agriculture. And once that receptor was exposed, a hidden evolutionary story became visible. Humans carry a heat-sensitive ion channel inherited from deep vertebrate history. Capsicum plants evolve molecules capable of manipulating versions of that channel. Birds, with different receptor sensitivity, could consume pungent fruits without the mammalian burn. Humans, despite possessing the susceptible mammalian machinery, refused to follow the avoidance behavior the plant chemistry encouraged. We domesticated the deterrent, then we trained our own sensory systems to tolerate it. So is your tolerance for spicy food hardwired into your ancient genetic code? Partly. Your genome helps build the receptors and signaling pathways that determine how the first encounter feels. Genetic variation can measurably alter capsaicin and heat sensitivity, and modern studies continue to identify variants associated with different tolerance-related traits. But your DNA does not write the final menu. Repeated exposure can desensitize the capsaicin response. Culture can normalize the burn. Learning can convert warning into reward. Personality and expectation can influence whether intensity feels thrilling or unpleasant. PubMed. The strongest scientific answer, therefore, sits between biological destiny and pure choice. You were born with an ancient fire detection system. You were not born with a fixed decision about whether to enjoy setting it off. Every hot pepper places those two histories in direct contact. Millions of years of sensory evolution and a few years of personal experience. The first history built the alarm. The second teaches you what the alarm means. And that is why the person across the table can bite into the same pepper, receive a different intensity, carry a different memory, and make a completely different decision about taking another bite. The chili is chemically the same. The nervous systems are not. Neither are the lives that trained them. Your love of fire is not simply encoded in one gene, nor is it merely a matter of willpower. It is a negotiation between an ancient receptor, a plant's evolutionary defense, and a human brain capable of learning that pain does not always mean danger. Long before the first cultivated chili entered a meal, TRPV1 was already guarding animal bodies from heat. Long after humans discovered peppers, that molecular guard still fires when capsaicin arrives. What changed was not the basic alarm. What changed was us. We learned to hear an ancient warning and call it flavor.